Okay, I'm not very sure how far I'm going to get with the third part of vision here on Wednesday. So I'm going to put color vision here, and we'll see if we get to talking about the visual pathways, as we really want to get this information to the brain and to the cortex for higher level processing. But let's finish up with color vision. So I like to leave this in here, even though I'm usually behind at this point, but I find these species differences really fascinating, and it always reminds me to say a few things. I hope other people find this as interesting as I do, but so we have these three color mechanisms, as trichromatic color vision theory suggests, these three different cones. If we look across species at how many cones different animals have, mice are monochromats. Cats and squirrels are dichromats, dogs also, so a lot of our mammals are dichromats, and some monkeys are, but New World monkeys. So what happened during evolution was that there was a mutation in the two cones that we had, the either long or medium wavelength, I can't remember which one, had a mutation to where sometimes there were these two, and that mutation was good for survival, because if there were the two, if you genetically got that mutation, you could see a wider range of colors. So we have New World monkeys are trichromats as well as goldfish. For some of those New World monkeys, that mutation allowed for them to see the difference between ripe bamboo and bamboo that wasn't ready, so exactly the specific food that they ate. But for us, the differences in colors of fruits and vegetables and everything, you can see how much that would help survival. And then we do see honeybees and birds are tetrachromats, so they have more cones than we do. If you remember me saying when we were talking about the electromagnetic spectrum that they were seeing ultraviolet light as well. So they have another cone. They are seeing a different part of the light spectrum. There are pictures out on the internet that try to give us some idea of what they are seeing, that are interpreting the ultraviolet light for us. Obviously, it's not going to be exactly what they're seeing, as we can't really experience that, but it's really neat as these flowers that we see as totally yellow, for them, have this kind of bullseye that is the brightest part of the bullseye is telling them that this is where their food is, and it's just really cool to see. And interestingly, so I think I got this from an old, I don't know if this is in this version of the textbook anymore, but people have different distributions and ratios of these different cones. And as far as we know, this doesn't translate into really differences in perception. But so I kind of kicked myself for not saying it with the tetrachromats on the last slide, but I can say it here, that it appears that we do have some human individuals who are tetrachromats. And from what I understand, they are women. As far as I know, they're only women, but they're at least typically or usually women. And they are those people who they have some kind of anomalous trichromat back in their genetics, but that's just on one of their X's, and then on their other X they have the normal. So they'll end out with three cones up in that medium long range, and they say they see colors as sort of more. They see a larger array of colors than we see, which I think is neat. Again, as far as we know, this basic difference in distribution and ratios, that's not really affecting the individual differences in color perception. I often get into discussions with students about whether or not we are seeing the same red when we are pointing to red. We've all learned to point to certain colors and to name them. This is red, this is pink, this is brown. But whether or not we're seeing those colors exactly alike, we don't know. All we know is that we can agree on the names. We might be having really different experiences with color. And there are still questions that and phenomena that trichromatic theory of color vision cannot explain and cannot answer. So I'm going to talk about another theory of color vision. The opponent process theory of color vision was really proposed originally by Ewald Hering back in the early 1900s. I want to say it was a little bit earlier than even that, but that these two theories of color vision, at the time we had behavioral support, but there was an argument about which one, whether it was trichromatic or whether we had these opponent processes. But we're going to see the evidence is showing that they're both in play here. So according to Hering, we have 4 primary colors, and they act in this kind of antagonistic way, or in this opponent way. And those antagonistic pairs are red-green and blue-yellow. And he also, because of when he was working, had only behavioral support of these opponent responses. He looked at negative color after images. So I'm going to show you one of these negative after images. If you're looking at the fixation cross and you're looking here for about a minute, I'm going to leave this up here for a minute. I'm not sure what really to talk about during that minute, so I'm mostly going to leave it up here in silence, but you really need to keep your eyes on the fixation cross for this to work. If you keep your eyes there, I'm looking at my timer every once in a while. I haven't actually done this in a long time because usually I'm at the front of the classroom. But remember, red-green is one antagonistic pair, blue-yellow is another, and I'm going to go ahead and put in there black-white. So it's been about a minute. If you can tell what you see, I hope it worked. I'm not sure if it's your full screen. For me, it was a part of a screen. And again, I kept looking at my timer and kind of doing something else, but you really have to look at it to, what we're doing is fatiguing those neurons that are responding to, in that case, I get my red, white, and blue, green, black, and yellow, so that we get that opponent response. And hopefully you are seeing a red, white, and blue flag at this point. So again, if you see how long ago he was doing his work, it was a good at least 60 years or so before we received physiological support for opponent process theory, where we just had the behavioral evidence before. They did this with single cell recording from retinal ganglion cells and from cells in the lateral geniculate nucleus, so in that part of the brain and the thalamus, which provided this physiological support, where they found opponent neurons. And so I'm just going to explain the left-hand side, where it says B+, yellow minus, of the left-hand picture to start with. So if we look at spontaneous firing, so I'm not looking at blue or yellow, that's spontaneous firing. If I show this neuron, or if I'm recording from this neuron and I show the person a blue wavelength, so 450 nanometers, we see that it gets excitatory. So that B+ is a blue excitatory. If I move up into 510 nanometers, which is going to be green, we see what looks a little bit like spontaneous firing. If I move up into 580 nanometers, so what is yellow, you can see that's inhibition, right? The neuron is not firing. We move farther up into red, so 660 nanometers. It also looks like there's some amount of inhibition, but not quite the amount of inhibition that we see in the yellow. And so we see a similar kind of pattern in the R plus G minus, except now it's responding to the red, and it's being inhibited by the green and maybe a little bit by the yellow. And what you can see on the right-hand picture, what's trying to be shown here, is those cones that are responding to a particular bipolar cell that's going to communicate to the ganglion cells. And between that, one of those horizontal cells that is allowing for, remember, lateral inhibition, where I can have my excitatory synapses and inhibitory synapses where the, let's say, the long and medium wavelength cones are excitatorily communicating to the horizontal cell, and it is sending inhibition to the bipolar cell, whereas the short wavelength cone is sending excitatory information to the horizontal cell, but also directly synapsing, so sending excitatory information to that bipolar cell. Okay, so I have this blue excitatory or short wavelength cone excitatory, long and medium wavelength cone inhibitory, which is how we get the B plus yellow minus outcome. And just so we see this again, look at here, we are again with our center surround receptive fields. I know you wanted to come back to it. So we have red, we can have red on, green off, red in the center on, red in the center off, green on in the center, red off, green off. So inhibitory center, excitatory surround where the red is in the surround in this case. We have all of those possible combinations. In this particular, so Gouros 1968, I think is where they found blue excitatory in the middle, yellow inhibitory in the surround. What I remember is that I think later on they found these different possibilities that we see in the red-green with blue-yellow, but some people say that's not so, but I think those papers were earlier. I'm not promising on how that works because I often forget, but different people are saying different things. But we can see all these different combinations, certainly for the red-green, maybe for the blue-yellow. So for some classes, I show this and do the same thing. For some classes, I don't. So if you enjoy these illusions, I recommend strongly that you do this one, that you're looking at the X in the center. Opponent process theory does not explain this illusion and the negative color after image. If it works for you, just so you know, it does not work for me. It does not work for all students. Every time I do this, I have different students having really different experiences that when I do this for a minute, again, you're going to want to pause this for another 30 seconds or so and just look at the X in the middle. I'm going to continue on. And so for some of us, that square became a whole square, and so those places where the circles, those black circles were entering into it, don't enter into it. And so we know that something is going on at the level of interpretation and that we're seeing that being filled in at the cortex. We see the same thing for lateral inhibition. Usually in sensation and perception, I go through several examples of how lateral inhibition at the level of the retina is not explaining some of our visual illusions, and some of the times we don't get visual illusions. So something's going on at the cortex as well, where we are interpreting that image. Some people have the full square, some people don't. I don't. If we ask which theory of color vision is right, which one's correct? Well, both, right? Because trichromatic vision, color vision theory is explaining what's happening at the level of the photoreceptors. We have 3 kinds of photoreceptors that respond differently to different wavelength information. And opponent process theory, that's describing what happens at a later level because of the integration of those inhibitory and excitatory signals. So at the level of the retinal ganglion cells, after we've had that convergence and inhibition, those processes, as well as what's happening at the lateral geniculate nucleus. I'm going to go ahead and tell you that we have in the cortex some opponent cells, which is why some people are doing that kind of interpretation at the level of the cortex. We still have opponent mechanisms there, or neurons that are responding in this opponent kind of way. There are limitations to these theories of color vision, even though they're explaining a great deal of how color vision occurs, but both the trichromatic theory and the opponent process theory have limitations. And the most important limitation is the fact that they don't really explain color constancy very well. So this ability to recognize color despite changes in lighting. So if I'm wearing, let's just say, my 510 nanometer monochromatic shirt, out in, I don't have that, out in the sunlight, that sunlight, that what's being emitted there in the lighting is across the electromagnetic spectrum. But if I walk inside and I'm under these kind of tungsten lights, where what they are emitting is more long wavelength light, then what's being reflected has been shifted over to those long wavelength lights because of what's being emitted. And if I'm walking into those ones that we now see, the LED lights that have more short wavelength light being emitted, again, I might be shifted over more towards the blue end of the spectrum because what I'm reflecting is necessarily influenced by the lighting that I'm under. And yet, when people walk inside with their green shirt, it doesn't appear to change into a yellow shirt suddenly to us. And so this color constancy is difficult to explain with just trichromatic theory and opponent process theory. Retinex theory is the theory of color vision that attempts to explain this by suggesting that at the level of the cortex is comparing information from various parts of the retina to determine the brightness and color for each area. So to some extent, I have the influence of just the memory of the fact that things like bananas are yellow are going to help me to interpret that. But if I'm seeing a banana amongst oranges and apples and so forth, those other colors that are surrounding that yellow, we're comparing those various parts of the visual field of what's happening at the retina in order to see that, yes, the banana is yellow. And this shows the importance of context in the world of vision and for our interpretation of color. And so I'm going to talk about this as the context of color perception, even though these pictures from the publisher, they are just wrong and they just do not work. This is on page 157 of your textbook, and he is showing these kind of Rubik's cubes under a yellow light and a blue light, but what he's doing that is very cool in the textbook, that's not at all what they're showing here, is he is on the left-hand side pulling out those blue squares. You see those 4 blue squares, and showing what that looks like without context of just what is being shown into our eyes from those blue squares under the yellow light. And then under the blue light, he pulls out the yellow squares. And so those yellow squares taken out of context. And what it turns out is the blue squares on the left-hand side. So this does kind of work to some extent, except you can't really see. There aren't clear yellow squares. Those are gone from the publisher's pictures. Yeah, they kind of look beige. Okay, those beige squares, you can tell in the publisher's pictures that they are. I mean, in the textbook, that those are yellow. That those, when we pull those out, those are actually, they appear exactly the same when we don't have the context. But when we do have the context, the blue appears blue and the yellow appears yellow, and we really, to some extent, pull this out from the light, the illuminating light, the yellow light or the blue light. And similar to color constancy, we have a brightness constancy that your author talks about very briefly here, and I'm only really talk about it because I really enjoy visual illusions. So if you're making this your whole screen and listening to me, you can put your finger so that, okay, so most of these you can see, let's go to the one on the right, the bigger one that you can actually see on the right, that object that looks folded over, the right-hand side is darker than the left-hand side. The left-hand side is pretty white, right? And the right-hand side is kind of gray. And that appears to be happening in this one in the center. But part of what's happening is that we are interpreting that white part, what appears to be white, as being in shadow, and the upper part as being under the light. So all you have to do is put your finger up and block out where it shifts, or where the change is, where the bend is, and you can see that those are exactly the same color, or the same amount of gray, the same amount of light being reflected into your eyes. They are the same shade. But once you pull your finger away, again, you can't stop having the visual illusion that that is something that is white, that's under shadow, and something that is dark, that is under light. This is a kind of brightness constancy of how we interpret information that is under shadow. And again, it's similar to color constancy, that we are doing this kind of interpretation that is based on context. I'm going to finish up with color perception, and then I'm not going to have enough time to move on into visual pathways. I'm now a little bit more behind than I want to be, and I always take a little bit longer than I want to on this first part of vision because it's complicated and because color perception has kind of a lot to it that I think is interesting. But so I hope we're going to move through the brain pretty quickly on Friday. But so color perception depends on both the light reflected from an object and how it compares with those surrounding objects, as we were seeing this kind of color constancy. It looks like area V4, so I'm going to talk about the primary visual cortex as being V1. As we're getting into our cortical processing of what's happening in the cortex, this higher level processing area called V4 appears to be what's responding to a variety of wavelengths and is apparently responsible for color constancy. And so I know in section 1 we started talking about whether or not people who are color blind, that was always what was happening in the cones, or whether that could be something that was happening in the brain. Here is an example of, so people who have damage to this area V4 get what's called a cerebral achromatopsia. And depending on the amount of damage, they might just have problems with color constancy. But as we move out a little bit from V4 as well, we see they end up having a monochromatic world. They are seeing the world without color. There's no easy specific place where we say this is where color vision happens. We're kind of putting it here in V4, but if you're really interested in this kind of thing, I know that last semester we ended up reading an article. It was a little bit more complicated than I wanted it to be, so I might try and find another one, but it was a really interesting kind of examination of this kind of color blindness, damage to different areas of the brain, and how that translates to differences in our ability to perceive color.